In this video, I'm going to talk about the gut microbiome, and in particular, the small intestine gut microbiome. I'll make a different video on the colon, the colonic microbiome in the future after I get finished with videos talking about the small intestine. And so the small intestinal gut microbiome is quite a bit less well understood than even the colonic gut microbiome, which is itself, still an active area of research. But what we do know about the small intestine microbiome is that there's fewer bacteria and fewer there's less diversity of bacteria there. And so in both of these images, which both come from different papers, you can actually see, so this one on the right, you can kind of see they have it colored in by how much bacteria there is in the intestine. So you can see in the small intestine, as you go through, it sort of gets more and more populated by bacteria. And then when you get to the large intestine over here, that is, quite a bit more populated by bacteria. And we see that over here as well. So the duodenum is on the order of 10 to the 3, the jejunum 10 to the 4 to 10 to the 5, the ileum 10 to the 7 to 10 to the 8, and the colon is up to 10 to the 12. And so the small intestine has less bacteria, but also as you go through it, it does get more and more as you make your way through from the duodenum to the jejunum to the ileum through the small intestine. And so over here, so we can see some of the dominant gut phyla. So if you remember your biological classification, kingdom, phylum, class, order, family, genus, and then species. And so we can see some of these small intestine families here, and some of the colon families here. And you can see that these are predominant, obviously, in both places, especially the colon. There are just a huge variety of different bacteria in there. A lot of them probably haven't even really been classified or characterized yet because they're sort of difficult to isolate. But these are some of the predominant ones. And so we see even among these, the colon is more diverse. So in addition to having just a greater number of them, it also has a greater diversity of them. All right, and so we can go down here, and this is just sort of looking at, so both in the small intestine here and in the colon here. So the small intestine has this mucus layer that's a bit more uniform than in the colon, which has this sort of outer loose layer here. And so these mucus layers sort of determine like what sorts of bacteria can live in them. So like the mucus is very difficult for the bacteria to make their way through. So only bacteria essentially that can break down mucus are able to get into it. And I think they have what's called mucinogen that can break through it, because the human colon, or the human intestine in general, has things in it that actually look for bacterial flagellum and attack those things. And so being motile is actually sort of selected against in the human intestine. Some bacteria do still have a flagellum. I think the Helicobacter pylori and things like that have a flagellum. Right, so several factors limit the ability of the gut bacteria to access host cells, including the mucus layers in the small intestine, the colon, antimicrobial peptides in the small intestine, including those by the Paneth cells, which I've talked about before. So also the steep oxygen gradient, so there's more oxygen sort of down lower, and so anaerobic bacteria are not going to be able to live down there. The surface of the small intestine is shaped into villi crypts and is colonized by certain adherent species, so species that can sort of adhere into the mucus and even onto the layer or onto the cell layer there, including segmented filamentous bacteria, SFB, lactobacilli, and Helicobacter, and then SPP just means sort of multiple species. And so, yeah, there's some bacteria that can live in the mucus. Most of them live sort of outside the mucus. And so that's where most of those will be found. But some of these who can live in, higher oxygen environments and with, a lot of these sort of immunoglobulins and things like that, and just in the thick environment of the mucus, will be able to live in there. And so I also found this over here, which is this huge sort of well, I think it's actually more of a genus tree. I was going to call it a family tree, but so this is looking at so obese and normal weight individuals represented by red and blue nodes respectively, and the size of the node is sort of how much they found in their sample here. And so you can see that even the sizes of these kind of vary quite a bit between all these different genera here. And so the abundance of them, and essentially what I wanted to show with this, because I don't really want to go through every genus in here, because it'd be kind of boring and probably wouldn't be that helpful, is that that these populations can change fairly rapidly. Just based on like your diet, that's why like the obese people have sort of differences compared to the normal weight people, but also just the bacteria in your intestine will change just depending on sort of what you recently ate. So I think it can change like within like as fast as like within a day or so. And so, yeah, like I said, this is just a bunch of these different genera here. So this is another thing that's showing sort of the phyla in the genera here, using the either standard diet, high protein diet, high fat diet, or high sugar diet. So as far as phyla is concerned, one thing to notice is that this Bacteroidetes here is much higher in the stool samples here. So this fecal samples is stool samples, and then mucus samples is from the small intestine. Then we can look at the genus or the genera over here, and so you can see between like the standard diet, the high protein, high fat, and high sugar, that you're getting these sort of changes in the population proportions of these different genera here. And we have that over here as well. So this is looking once again at obese individuals and normal weight individuals. And so I boxed some of these because these were the ones that were like predominant in the small intestine here. And so you can actually see this Firmicutes. And by the way, this is phyla here. And then these proteobacteria are pretty predominant. And this actinobacteria here, which is this kind of color right here. And so once again, going through each one of these phyla or genus and sort of, naming them and, saying, ooh, look at that, that's the proportion. I'm more going for an overall thing here that these population proportions can change relatively quickly. All right, and so, and down here, so this is sort of looking at fatty acid beta oxidation, which is the breakdown of fatty acids. And so there are more bacteria in the small intestine uh, that can undergo this, or that have increased fatty acid beta oxidation capabilities when you have a high fat diet. And so uh, I'll just read off what I have here on my notes. So the small intestine microbiota differ when comparing mice on low-fat and high-fat diets. A high-fat diet induces an abundance of Clostridia, while decreasing the bifidobacteria and bacteroides, creating a microbial genetic profile with an enhanced capacity for inducing genes involved in small intestine epithelial lipid transport and pancreatic cholecystokinin signaling. Indeed, when the high-fat diet microbiota was transplanted into germ-free mice, lipid absorption improved. We know from fecal-based human studies that diet can alter the colonic microbiota in metabolites within 48 hours, as well as lead to long-term reductions in the diversity and taxonomic changes, such as increase in the Firmicutes and proteobacteria. So the gut microbiota of individuals who are obese seem to have less bacterial richness and increased level of Firmicutes and reduced level of bacteroidetes. And so essentially, this is just talking about how how there are these changes in the bacterial populations based on what kind of diet you have, and that there is sort of the high fat diet ends up having more sort of fat absorption. Um, so I also found that, um, so where was that? So yeah, processes such as sugar phosphotransferase systems, the pentose phosphate pathway, and lactate propionate fermentation, as well as cofactors such as cobalamin, which is vitamin B12, and biotin, were encoded across many taxa from ileal effluent, and particularly Streptococcus, arguing that carbohydrate metabolism is a central function of the collective small intestine microbiota. This contrasts with colon communities, which are more equipped to degrade complex carbohydrates. Uh, and so, what they find is that the small intestine bacteria, like they said, is more focused on carbohydrate breakdown, whereas so simple sugar breakdown, I guess you could say, things like glucose and sucrose and lactose and things like that, where the colon would be more, sort of like fiber type things. Um, and so the small intestine, as I said, is more focused on the sugar breakdown. So biochemically, bacterial fermentation of carbohydrate sources leads to production of organic acids, aldehydes, alcohols, and gases. When excessive fermentation occurs in the small intestine, metabolic byproducts contributing to bloating, nausea, abdominal pain, distension, and acidic stools. Typically, primary bile salts assist with fat absorption before deconjugation. So the bile salts, as I talked about in the previous video on the sort of physiology, sort of, make the sort of bubbles of fat that are sort of suspended in the water smaller. And so if we are deconjugating these bile salts, then we are going to, so the primary bile salts assist with fat absorption and before deconjugation and reabsorption in the ileum. With bacterial overgrowth, however, steatorrhea, or fatty stool, and fat-soluble vitamin deficiency can result from premature bacterial deconjugation of primary bile salts. So, in other words, the bile salts sort of detach from these sort of lipid droplets in the chyme or in the the food processing, and so that can lead to well, it slows down sort of fat absorption and can create this thing called it's steatoria or steatoria, which is essentially just fatty stool. Your feces is more fatty and can reduce the fat soluble vitamin intake. And so these bacteria all have been shown to synthesize um, vitamin K as well. And so small intestinal bacterial overgrowth, or SIBO, maybe you've heard of that or have even suffered from it, is a condition associated with impaired vitamin K metabolism in human beings. Vitamin B12, whose digestive physiology is intimately linked with the small intestine, it also is impacted by bacterial biology. And essentially, what it's saying is that, a lot of these different vitamins, if you have this condition called small intestinal bacterial overgrowth, then you can end up actually getting less of these vitamins absorbed. And, this can result from things like overeating and, other, I guess, bacterial infections and stuff like that in the small intestine. But anyway, so again, I've really just kind of scratched the surface on this small intestine microbiota. I'll probably go into a bit more detail on when I get to the colon and the colonic microbiota, just since there's quite a bit more known about that. It was hard for me to find really good information, and even harder for me to find good sort of figures like this, which is why this video was a lot of me just sort of reading things off of my notes here rather than going through figures. Uh, but yeah, I hope this gives you at least some appreciation for the fact that the while the colonic microbiota sort of gets all the press, there is a microbiome in the small intestine as well, and it can be involved in various pathogeneses and things like that. And so, once again, as usual, there will be references in the description down below. So if you're interested in getting an even closer look at some of this stuff, I would highly recommend that. I'll put my lecture notes down there as well, so you can study those a little bit closer if you didn't catch everything I said in this video. But anyway, I hope you, like I said, have a better understanding, or at least a better appreciation for the small intestine microbiome. And I will see you in the next video.